Our story begins at a breast cancer screening camp in 2021. It was at this point that we realized that there is a high prevalence of breast cancer cases in a local community of Vidura, Kerala. We found the same troubling trend in cases globally as well, with worldwide projections of around 3 million new cases and 1 million deaths by 2040. In an attempt to look into the existing therapies, we spoke with the breast cancer survivors who shared with us their struggles associated with the existing cancer drug therapies. Cancer cells do overexpress cell markers, and we are making use of this property in targeted drug therapy. So, only if a cell overexpresses a particular marker will the drug be delivered, inducing cytotoxic action. However, there is a basal level of expression of these markers in normal cells as well, leading to off-site targeting. Now, the question is whether we could build upon targeted drug therapy to get a better and an efficient drug delivery mechanism. We came up with a solution, Duongo. Good day everyone, this is team IISER TVM presenting to you Duongo, a dual nanovesicle drug delivery system targeting breast cancer. Duongo, as the name suggests, a dual targeting mechanism. Now the immediate question that pops up is, what can be a drug carrier of choice? We chose bacterial outer membrane vesicles or OMVs, essentially nanovesicles produced by gram-negative bacteria. So these can be loaded with a diverse set of payload and they are inherently immunostimulatory, which also aids into tumor growth inhibition. Now the best part of outer membrane vesicles is that their surface can be decorated with affinity proteins of choice, allowing for targeted drug delivery. Under the suggestion of Dr. Bharat, we decided to work on HER2 positive breast cancer due to its poor prognosis. Now, we intend to work on a dual targeting system. But what are our targeting markers? We chose HER2 and CX3CR1 as both of them are overexpressed in HER2 positive breast cancer. Now here we have our drug carrier of choice, which is the outer membrane vesicle, and here we have our targeting markers. So how do we target these markers using outer membrane vesicles? There comes affinity proteins. Incorporating the suggestion of Dr. Vipul Gujarati, we decided to go ahead with Affibody, which is a small antibody mimetic against HER2 as the first affinity protein of choice. And incorporating the suggestion of Dr. Ferreira, we decided to work with tumor homing peptide or THP, which is a short oligopeptide against CH3CR1 as second affinity protein of choice. Now that we have our dual targeting system in hand, what is the drug that we are delivering? A Prodrug-Enzyme System. A prodrug is an active form of a drug which gets activated only in the presence of an enzyme. Now, let's take a glance at how it works.
We have a hypervasculating strain of E. coli, which essentially just means that they produce more number of vesicles than normal. We will then be transforming these E. coli with plasmid having the gene construct of our desired affinity protein. This protein will then preferentially localize to the outer membrane of the bacteria. From the outer membrane, the vesicles will start to bleb out, expressing our desired affinity protein on its surface. Now, we will have two types of OMVs. Type 1 OMV expressing the Affibody on its surface and type 2 OMV expressing THP on its surface. Now, we will be loading them with prodrug and enzyme simultaneously. Now we have our cancer cells overexpressing both the cell surface receptors. Only when both these two types of OMBs simultaneously internalize into our cells, then it will lead to conversion of prodrug into its active cytotoxic form, leading to cell death and tumor growth inhibition. In the case of normal cells having basal expression of either one or both receptors, it will lead to low or no internalization of OMBs, resulting in minimal side effects. Now, since we have designed our project, let's look into how we can build and test it.
As a pilot proof of principle study, we defined four goals for our project that we believed were essential to demonstrate the functionality of Duongo. The first goal of our project was to isolate native or unmodified OMVs from E. coli. Now, normal E. coli K12 strains that we have in the lab naturally produce OMVs, but the slow rate of production makes isolating OMVs from them a very resource intensive process. So, to overcome this, we acquired four hypervesiculating strains of E. coli that produced more OMVs than the parental K12 strain. Each of these strains had a single gene that had been knocked out. So, the four strains were the TOLA knockout, the TOLR knockout, the DECP knockout, and the NLP1 knockout. So how did we go about selecting one strain out of these four? We started out by constructing a growth curve to compare the fitness among these four strains, and we found that the DEC-P knockout showed delayed growth kinetics, and therefore we excluded it from the remainder of our project. Our literature survey informed us that the TOL-R knockout produces more OMVs than the TOL-A knockout. This left us with just two strains, the TOL-R knockout and the NLP1 knockout, and we wanted to see if there was a difference between the size distribution of OMVs yielded from the two strains. To answer this, we used the technique of dynamic light scattering to compare the yield of OMVs that we obtained from culturing both strains under identical culture conditions. And we found that there was no significant difference between the size distribution of OMVs from either TOL-R knockout or the NLP1 knockout. Finally, we compared the yield of OMVs between the two strains and found that the NLP1 knockout produced more OMVs than the TOL-R knockout under identical conditions, and therefore we chose the E. coli NLP1 knockout to be the strain of our choice for our Duonco project. Now, it's not merely enough to isolate OMVs, but we also need to experimentally verify their presence in the isolated fraction. For this, we use electron microscopy to image the isolated fraction, where we observe spherical structures that were bound by a lipid bilayer, therefore indicating that we had successfully isolated OMVs. One final part of the first goal of our project was to assess the presence of contaminants in our isolated OMV fraction. A problem that is commonly associated with OMV isolation is that bacterial cell components often co-sediment along with the OMVs, and this can be especially problematic if we plan to use the OMVs for downstream applications such as drug delivery, where these bacterial cell components can trigger an unwanted immune reaction in the patient. So for this, we analyzed the isolated OMV fraction using SDS page, where we noted bands roughly at the 35 kilodalton mark, which we believe are members of the outer membrane protein family, which is a group of proteins that are present in the outer membrane and hence OMVs. We noted a general absence of proteins at the other molecular weights, which informed us that bacterial cell contaminants were generally absent from our isolated fraction. With this, we concluded the first part of our project by selecting a hypervesiculating strain, isolating native OMVs, performing experiments to verify OMVs, and then confirming the absence of contaminants. The second goal of our project was to isolate the two types of engineered OMVs, the AFI OMVs and the THP OMVs. To begin with, we had to design our recombinant proteins. Our recombinant proteins had four segments. The first segment was cytolysine A, and this segment was responsible for localizing the entire recombinant protein to the outer membrane of the bacteria and hence to OMVs. The second segment was a flexible serine glycine linker that was responsible for linking the first segment and the third segment. The third segment was an epitope tag, the V5 tag, which we plan to use for immunodetection in Western blotting and cell microscopy experiments. The terminal section of our recombinant protein was the anti-HER2 affibody and was responsible for generating affinity against the HER2 breast cancer biomarker. Our second recombinant protein followed a similar structure, but the V5 tag was replaced by the 3X flag tag so that we could detect both recombinant proteins at the same time, and we replaced the anti-HER2 affibody with the THP protein, which is responsible for generating affinity against the CX3CR1 breast cancer biomarker. We obtained gene fragments coding for these two recombinant proteins, and we separately ligated them into E. coli expression vector PGEX4T1. We performed several experiments to confirm successful ligation and cloning. The first of which was a colony PCR, and we found several positive clones for both the affibody and the THP protein. Similarly, we obtained a positive restriction digestion with inserts at the expected band weights. Finally, we performed Sanger sequencing to sequence our inserts and verify that they were free from any mutations. Now, because our project involved expressing recombinant proteins using IPTG, we asked the question, what would be the optimal concentration of IPTG to use to induce our bacterial cultures? For this, We used math modeling and constructed a gene regulatory network consisting of all the relevant molecular species for the process, and our predicted results told us that at approximately 1 millimolar of IPTG, we would obtain optimal protein expression, and we used this value going forward. Next, we wanted to test the expression of both the Affibody protein and the THP protein, so we cultured strains harboring these plasmids and then induced them using IPTG. We analyzed these cell pellets using SDS page, and we noted the expression of both the Affibody protein, which is indicated here in green with the green arrow, and the THP protein, which is indicated here with the red arrow. To further verify the expression and the localization of our recombinant proteins, we used Western blotting. We detected the presence of the THP protein in the bacterial cells following IPTG induction, and we also noted its presence in the THP OMV fraction. This indicated that the THP OMV actually contained the THP protein. Similarly, we noted the expression of the Affibody protein in the cells following IPTG induction, and we noted its presence in the Affi-OMV fraction, indicating that the Affibody protein had successfully localized to the OMV fraction. Now, while blotting against the V5 tag, we picked up on a lot of background, and we also noted expression for both proteins in the cells before IPTG induction, which we think can be explained by the leaky nature of the lac operon. But we would like to further replicate these results with more experiments. This concluded our second goal, where we successfully isolated both types of engineered OMVs. Now, the third goal that we believe was crucial for our project was to assess the safety of these engineered OMVs. For any drug delivery system, it's important that the drug delivery carrier itself is not toxic, but that the toxicity is derived from the payload. And we want the same to be true for our project. To investigate this, we incubated the breast cancer cell line SKBR3 either with PBS or two different concentrations of the THP OMV, and we measured cell viability using the MTT assay. Our results indicated that the THP OMVs are not significantly cytotoxic, and this was encouraging evidence. The final goal of our project was to actually see if these engineered OMVs were capable of internalizing in a target breast cancer cell. For this, we used the breast cancer cell line SKBR3, which overexpresses both the target markers HER2 and CX3CR1. We devised three treatment regimes corresponding to native or unmodified OMVs that do not carry these recombinant affinity proteins on their surface, the Affibody protein against the HER2 protein, and the THP OMVs against the CX3CR1 cancer marker. Following incubation of these three types of OMVs, we imaged them using confocal microscopy. For the native or unmodified OMVs, we detected signal primarily from the extracellular medium and did not see much signal from within the cells, indicating that internalization likely had not occurred. For the SKBR3 cells that were incubated with the Affi OMVs, we detected the signal at the cell periphery, indicating that the OMVs had potentially bound to the cell surface and internalized. Finally, for the SKBR3 cells that were incubated with THP OMVs, we detected signal from the interior of the cell, indicating that internalization had occurred. These experiments indicate that our engineered OMVs, when compared to the native OMVs, were capable of internalizing in this breast cancer cell line. However, due to the low concentration of OMVs used, and due to the fact that we suspect that OMVs were aggregating in this medium, we could not conclusively conclude if internalization had occurred, but these results were definitely encouraging. The last important question we had was that what would happen if these engineered OMVs were to encounter a cell line or cells within the human body that did not overexpress these target markers. And so for this experiment, we used SKBR3, the breast cancer cell line from before, and HEK293T, a cell line that does not overexpress both target markers and therefore serves as a negative control. We incubated both types of cell line with both types of OMVs, and following incubation, we imaged them using confocal microscopy. When the SKBR3 cell line was treated with the Affi OMVs and the THP OMVs, we detected signal from both types of OMVs from the interior of the cell, indicating that both types of OMVs had potentially internalized in the cells. For the HEK293 T cells, we did not detect a similar signal from both the THP OMVs and the Affibody OMVs from the interior of the cell. Our results indicate that when these engineered OMVs encounter a breast cancer cell line that was expressing the target markers, they internalized. But when they encountered a cell line that did not overexpress these target markers, they did not internalize. Now, there are some limitations, as there were from before, given the low concentration and the aggregation of OMVs. We also have an additional, we also believe that we may have picked up some non-specific signal while imaging the HEK293 T cells. Nonetheless, overall, our experiments provide encouraging preliminary evidence that these engineered OMVs are indeed capable of internalizing in a target breast cancer cell and therefore can be used to deliver a prodrug enzyme system. We would now like to address some limitations associated with our project. First, sample size that we used for our internalization experiments was quite small. Second, while performing our incubation and cell internalization studies, we observed a lot of aggregation of OMVs and some possible non-specific signal during microscopy. We also did not have the chance to assess the safety of our Affi OMVs, although we did measure it for the THP OMVs, and we believe that result can be mapped on the Affi OMVs. Finally, we were not able to assess the ability of these engineered OMVs to deliver a payload to the target cancer cell. Some experiments that we would like to carry out in the future include studying the internalization of these engineered OMVs in different kinds of cell lines to see what would happen when they encounter a cell that has different kinds of cell surface markers. With any drug delivery system, it's important that we understand what happens when these engineered OMVs enter the cell. And so we would like to monitor the fates of OMVs by loading them with fluorescent dyes and then following them after internalization using live cell imaging. Given that the V5 tag generated a lot of background on our Western blot experiments, we would like to replace it with another epitope tag for better detection. Finally, for our system to work, it's important that after release, the prodrug and the enzyme meet within the cell, and we would like to investigate this process using a split GFP assay. With any synthetic biology project, it's important that we assess and address the biosafety risk associated with it. With any drug delivery system, duonco also has the potential for misuse, where it can be used to deliver a harmful payload to either a human or an animal subject. However, we believe that our project does not represent an increased risk for dual use above and beyond what is already publicly available in published literature about OMVs, drug delivery systems, cancer drug delivery system, and prodrug enzyme system. Additionally, OMVs are non-replicating and they require sophisticated equipment for their production, which again reduces the risk of misuse and the chances of spread. Given that the prodrug and enzyme combination is cytotoxic to human and mammalian cells, we avoid conducting any experiments with it in our project. One of the domains in our recombinant protein, cytolysin A, is associated with a harmful effect on mammalian cells, and therefore we met with an expert, Dr. Rahul Rai, who has been working with cytolysin A, and with his guidance, we created a mutated form of cytolysin A with a single point mutation that made it safer. And we hope that synthetic biology teams in the future can use the same mutated version. Next, we actually quantitatively measured the toxicity of our engineered OMVs and found that they did not cause significant cell death. We obtained all the relevant safety approvals, including the Institutional Biosafety Committee and the Review Committee on Genetic Manipulation. We followed out appropriate lab safety during our experiments, and we have responsibly carried out our human subject research. Finally, to raise awareness about the importance of safety in biology and beyond, we released a documentary on how lab waste is processed after it comes out from a research institution like ours.
The thoughts and the feedbacks from our stakeholders were crucial in shaping our project. Based on the inputs we received from all the stakeholders that we have identified and interacted with, we categorize them into four broad groups. First,
We interacted with cancer healthcare professionals to specifically learn about their experience as providers to the end users of cancer therapy. Dr. Kavita Ravi and the other doctors that we met from Regional Cancer Center Thiruvananthapuram talked about the problems faced by patients due to the existing therapies. They also highlighted the importance of a more specific system targeting HER2 positive breast cancer as it is a very aggressive form of cancer. Our interaction with Ms. Barbara Demorest, founder of Knitted Knockers, showed the sufferings of people undergoing mastectomy. These interactions reminded us that a more effective treatment strategy is necessary. We also interacted with experts in the field of drug delivery and cancer in order to decide on the various components of our project design. Our meeting with Dr. Deborah provided us insights about the functionings and potential of peptide in Duo-Onco. Dr. Vipul Gujarati helped us with gene construct and Dr. Santosh Kumar helped us in choosing epitome tags. We also got directions regarding the kind of cell lines that we can work on. Through these interactions, our project became more precise. To properly assess the extent to which our system can be implemented to our end users, we consulted with representatives from local bodies and pharma companies. Ms. Sankita Topiwala took us through all the stages a project has to undergo before reaching to people. We even got an opportunity to meet Mr. Anup Ambiga, CEO of Kerala Startup Mission, with whom we could discuss our project concept. Local Panchayat President gave us information regarding the existing government policies to increase detection tests for cancer. By building on these discussions, we could design our future experimental plan. During our conversations with breast cancer survivors, caretakers, and the members of the general public, they emphasized the need for clarity regarding the diagnostic facilities and the preventive measures available to them. Survivors explained the difficulties they faced during the period of treatment. They preferred treatment techniques with fewer side effects. We conducted a series of interviews with the general public and distributed pamphlets promoting breast cancer awareness. Now, to get a better understanding of the public's knowledge concerning breast cancer, we distributed print and online surveys in 5 languages. Dr. Harlal Madhavan, an expert in health economist, helped us to analyze our survey results. And our analysis revealed that there is a significant information gap about early detection of cancer. This prompted us to hold a breast cancer awareness talk and a screening camp for the housekeeping staffs of our institution under the guidance of Medical Officer Dr. Hema Francis and in association with Indian Medical Association. This campaign made us aware of the high prevalence of breast cancer in our region and the need for early detection knowledge. Hence, we also used theatre as a medium for our breast cancer campaign to reach a diverse audience and people of all gender and ages. The objective of Let's Face It with Courage was to raise awareness about the significance of self-breast examination. Our breast cancer awareness campaign taught us that it is essential to bridge the gap between the academia and the general public to communicate about synthetic biology and its numerous applications effectively. We made sure to integrate all these suggestions into our project and we were able to close the loop between what is designed and what is desired.
After closing the loop, we wanted to incorporate what we've learned so far and outline the way forward by IEI, improving our design, extending the scope of our project, and building a framework for real-world implementation. Improving our design would involve changing the production of our OMVs by incorporating steps such as density gradient ultracentrifugation and ultrafiltration to improve the yield of the OMVs. After determining the amount of OMVs that have to be used, we would monitor the levels of LPS and ensure additional safety by creating knockouts or reducing the amount of LPS present. Duonco's design allows it to be extended to treat any other type of cancer by changing the relevant target epitopes on the surface. This inherent flexibility in our concept also allows us to tailor it to the different expression levels of markers in individuals. Our next steps would involve comparing our system's efficacy and safety and the mode of administration through in vivo testing and mouse models respectively, following which we would move on to human clinical trials. After achieving the work we have outlined for our future, we believe that the unique features of our system would provide the edge over existing targeted therapies and be a cure for cancer. Thank you, and we would like to thank our sponsors, And most especially, we would like to thank our primary PI, Professor S. Murthy Srinivasula, and our secondary PI, Dr. Sandhya Ganesh.